The Helmholtz-Center for Infection Research in Braunschweig regularly awards the Paper of the Month. The prize honors an outstanding paper published by a PhD student or a postdoc. The awardee for July 2014 is Kendra Bussey, who works in the junior research group Virale Immunmodulation, led by Melanie Brinkmann at the HZI.
Kendra Bussey. I'm a postdoc in Melanie Brinkmann's research group at the Helmholtz-Zentrum. The group is Viral Immune Modulation, so we're interested in looking at how herpes viruses can evade the innate immune response, and also to learn more about innate immune biology. So initially what we looked at was the innate immune response upon infection with a herpes virus, murine gammaherpesviruses68. This is a model for two human viruses, Epstein-Barr virus and Kaposi-sarcoma-associated herpes virus, both of which can cause human cancers and also vary in their prevalence in the population. And what we saw with the murine gamma herpes virus is that it doesn't induce a very strong innate immune response upon infection. And so this suggested to us that the virus may actually be intentionally avoiding the innate immune response. And so we looked then at the response to some toll-like receptor agonists. So toll-like receptors are present on the cell surface and in endosomes, and they're able to detect pathogen-associated molecular patterns as an early detection system for the host. And so we found that in cells that are infected with murine gammaherpesviruses68, that they're not responding to stimulation of these receptors. So the virus is actually blocking these signaling pathways to control the innate immune response. And we also found that this is conserved in Kaposi-sarcoma-associated herpes virus. And then we looked at which proteins may be involved in this pathway, and we were able to find one protein that actually removes one of the toll-like receptors from the cell surface. So it's able to block the pathway by getting rid of the receptor. It's very interesting that the virus is actually able to do this, because normally this is a way that the host would be able to detect an invader and then control the infection. But if the virus is blocking this, then that helps explain how the virus is able to set up infection. And herpes viruses establish lifelong latency, and so they've had to develop a way to be able to get to the host and stay there. And so this may be one of the ways that they're able to do this. And so this would be something that we would want to target, potentially. So next we're looking at actually how the proteins are specifically doing this. So we're looking into the mechanism. Another study that we're doing at the moment is in collaboration with Juli and Ulrich Kalinka at the Twin Core, and we're looking at what exactly is important for the host to be able to detect murine gammaherpesviruses68. So the virus is able to escape detection, but we want to know what pathways would otherwise be able to detect the virus. And so this is a really interesting study at the moment. So we're interested in understanding how herpes viruses interact with the innate immune system and how the host responds.